Dinosaurs have been extinct for over 65 million years. Their fossilized remains provide important insights into the lifestyle and the process of evolution into modern-day birds, the direct descendants of theropod dinosaurs. In the northeastern region of China, several very well-preserved specimens of feathered species from the early Cretaceous area have been unearthed. It has long been a topic of controversy since the appearance of these fossils as to what role these feathers played for these creatures. In the article, Fossilized Melanosomes and the Colour of Cretaceous Dinosaurs and Birds, published 2010 by Zhang and colleagues, details an experimental analysis of some exceptionally well-preserved fossilized feathers to determine, firstly, the similarities between filament-like structures in modern birds, and secondly, their ultrastructure appearance. Two main types of feathers were observed in this experiment: pennaceous feathers belonging to early birds, which are characterized by a central shaft and lateral veins, and tegumentary filaments belonging to non-avian dinosaurs that resemble small downy feathers. The structures of these were observed using hundreds of samples from both the outer and inner surface of the fossils at a submicron level using scanning electron microscopy, the result of which provided evidence to support two conclusions. From each sample, there was evidence of melanosomes, which are organelles housing pigments, in the form of arranged oblate bodies similar to those found in modern bird feathers, as indicated by the arrows in this sample taken from a Confuciusornis. It was also possible to distinguish the colour of these feathers due to the different shapes of each type of melanosome. They were found to possess both eumelanosomes, denoted by longer arrows, which are associated with black-grey coloration, and phaeomelanosomes, which give red, brown, and yellow coloration. Evidence was also found to support that these structures were indeed melanosomes, and not partially degraded dermal collagen fibres or a fossilized film of decomposing bacteria. This evidence includes the arrangement of different types of melanosomes, which are characterized by their alternative shapes in distinct bands, which would not be expected in bacterial growth or if they were dermal fibres. Likewise, these structures are consistent with samples taken from further inside the fossil, where bacteria would not have been present. However, still, the origin of these microbodies is still disputed, as detailed in the article Melanosomes or Microbes, testing an alternative hypothesis for the origin of microbodies in fossil feathers, published in 2014 by Moyer and colleagues, in which the authors detailed evidence that supports that it is more likely that in fact these structures are microbes. The experimentation in this article uses the same method of SEM scanning, but also compared these with results obtained using field emission and transmission electron microscopy. Despite the lack of conclusiveness on this topic, it may be inferred from the results of the first article that due to the banding and colour patterns in fossilized feathers, it is more likely that these developed as a means of sexual selection before further evolving into a key innovation, leading to the ability to glide and further on to power flight.